Section 46 of Anatomy of the Human Body, Part 5. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Anatomy of the Human Body, Part 5 by Henry Gray. The Spleen. Lion. The spleen is situated principally in the left hypochondriac region, but its superior extremity extends into the epigastric region. It lies between the fundus of the stomach and the diaphragm. It is the largest of the ductless glands, and is of an oblong, flattened form, soft, of very friable consistence, highly vascular, and of a dark purplish color. Development. The spleen appears about the 5th week as a localized thickening of the mesoderm in the dorsal mesogastrium above the tail of the pancreas. With the change in position of the stomach, the spleen is carried to the left, and comes to lie behind the stomach and in contact with the left kidney. The part of the dorsal mesogastrium which intervened between the spleen and the greater curvature of the stomach forms the gastrosplenic ligament. Relations. The diaphragmatic surface, facies diaphragmatica, external or phrenic surface, is convex, smooth, and is directed upward, backward, and to the left, except at its upper end, where it is directed slightly medialward. It is in relation with the under surface of the diaphragm, which separates it from the 9th, 10th, and 11th ribs on the left side, and the intervening lower border of the left lung and pleura. The visceral surface is divided by a ridge into an anterior or gastric and a posterior or renal portion. The gastric surface, facies gastrica, which is directed forward, upward, and medialward, is broad and concave, and is in contact with the posterior wall of the stomach, and below this with the tail of the pancreas. It presents near its medial border a long fissure, termed the hilum. This is pierced by several irregular apertures, for the entrance and exit of vessels and nerves. The renal surface, facies renalis, is directed medialward and downward. It is somewhat flattened, is considerably narrower than the gastric surface, and is in relation with the upper part of the anterior surface of the left kidney and occasionally with the left suprarenal gland. The superior extremity, extremitus superior, is directed toward the vertebral column, where it lies on a level with the 11th thoracic vertebra. The lower extremity or colic surface, extremitus inferior, is flat, triangular in shape, and rests upon the left flexure of the colon and the phrenico-colic ligament, and is generally in contact with the tail of the pancreas. The anterior border, margo anterior, is free, sharp, and thin, and is often notched, especially below. It separates the diaphragmatic from the gastric surface. The posterior border, margo posterior, more rounded and blunter than the anterior, separates the renal from the diaphragmatic surface. It corresponds to the lower border of the 11th rib and lies between the diaphragm and the left kidney. The intermediate margin is the ridge which separates the renal and gastric surfaces. The inferior border, internal border, separates the diaphragmatic from the colic surface. The spleen is almost entirely surrounded by peritoneum, which is firmly adherent to its capsule. It is held in position by 2 folds of this membrane. One, the phrenico-lional ligament, is derived from the peritoneum, where the wall of the general peritoneal cavity comes into contact with the omental bursa, between the left kidney and the spleen. The lional vessels pass between its 2 layers. The other fold, the gastrolional ligament, is also formed of 2 layers, derived from the general cavity and the omental respectively, where they meet between the spleen and the stomach. The short gastric and left gastroepiploic branches of the lional artery run between its 2 layers. The lower end of the spleen is supported by the phrenico-colic ligament. The size and weight of the spleen are liable to very extreme variations at different periods of life, in different individuals, and in the same individual under different conditions. In the adult, it is usually about 12 centimeters in length, 7 centimeters in breadth, and 3 or 4 centimeters in thickness, and weighs about 200 grams. At birth, its weight, in proportion to the entire body, is almost equal to what is observed in the adult, being as 1 to 350, while in the adult it varies from 1 to 320 and 400. In old age, the organ not only diminishes in weight, but decreases considerably in proportion to the entire body, being as 1 to 700. The size of the spleen is increased during and after digestion, and varies according to the state of nutrition of the body, being large in highly fed and small in starved animals. In malarial fever it becomes much enlarged, weighing occasionally as much as 9 kilos. Frequently in the neighborhood of the spleen, and especially in the gastrolienal ligament and greater omentum, small nodules of splenic tissue may be found, either isolated or connected to the spleen by thin bands of splenic tissue. They are known as accessory spleens, lion accessorius, supernumerary spleen. They vary in size from that of a pea to that of a plum. Structure. The spleen is invested by two coats, an external serous and an internal fibroelastic coat. The external or serous coat, tunica serosa, is derived from the peritoneum. It is thin, smooth, and in the human subject intimately adherent to the fibroelastic coat. It invests the entire organ, except at the hilum and along the lines of reflection of the phrenicolienal and gastrolienal ligaments. The fibroelastic coat, tunica albuginea, invests the organ, and at the hilum is reflected inward upon the vessels in the form of sheaths. From these sheaths, as well as from the inner surface of the fibroelastic coat, numerous small fibrous bands, trabeculae, are given off in all directions. These, uniting, constitute the framework of the spleen. The spleen therefore consists of a number of small spaces or areolae formed by the trabeculae. In these areolae is contained the splenic pulp. The fibroelastic coat, the sheaths of the vessels, and the trabeculae are composed of white and yellow elastic fibrous tissues, the latter predominating. It is owing to the presence of the elastic tissue that the spleen possesses a considerable amount of elasticity, which allows of the very great variations in size that it presents under certain circumstances. In addition to these constituents of this tunic, there is found in man a small amount of non-striped muscular fiber, and in some mammalia, for example dog, pig, and cat, a large amount, so that the trabeculae appear to consist chiefly of muscular tissue. The splenic pulp, pulpa laenis, is a soft mass of dark reddish-brown color, resembling grumous blood. It consists of a fine reticulum of fibers, continuous with those of the trabeculae, to which are applied flat, branching cells. The meshes of the reticulum are filled with blood, in which, however, the white corpuscles are found to be in larger proportion than they are in ordinary blood. Large rounded cells, termed splenic cells, are also seen. These are capable of amoeboid movement, and often contain pigment and red blood corpuscles in their interior. The cells of the reticulum each possess a round or oval nucleus, and like the splenic cells, they may contain pigment granules in their cytoplasm. They do not stain deeply with carmine, and in this respect differ from the cells of the Malpighian bodies. In the young spleen, giant cells may also be found, each containing numerous nuclei or one compound nucleus. Nucleated red blood corpuscles have also been found in the spleen of young animals. Blood vessels of the spleen. The lienal artery is remarkable for its large size in proportion to the size of the organ, and also for its tortuous course. It divides into 6 or more branches, which enter the hilum of the spleen and ramify throughout its substance, receiving sheaths from an involution of the external fibrous tissue. Similar sheaths also invest the nerves and veins. Each branch runs in the transverse axis of the organ, from within outward, diminishing in size during its transit, and giving off in its passage smaller branches, some of which pass to the anterior, others to the posterior part. These ultimately leave the trabecular sheaths and terminate in the proper substance of the spleen in small tufts or pencils of minute arterioles, which open into the interstices of the reticulum formed by the branch sustenticular cells. Each of the larger branches of the artery supplies chiefly that region of the organ in which the branch ramifies, having no anastomosis with the majority of the other branches. The arterioles, supported by the minute trabeculae, traverse the pulp in all directions in bundles, pencillae, of straight vessels. Their trabecular sheaths gradually undergo a transformation, become much thickened, and converted into adenoid tissue, the bundles of connective tissue becoming looser and their fibrils more delicate, and containing in their interstices an abundance of lymph corpuscles. W. Mueller. The altered coat of the arterioles, consisting of adenoid tissue, presents here and there thickenings of a spheroidal